My name is Gregory Lanza. I'm a cardiologist here at Washington University. I'm the principal investigator and co-inventor of a new product called Thrombosynth that was developed to detect fibrin clots in mechanical assist devices, and I am the co-founder of a small company to develop this called Capella Imaging. Probably 60 to 100,000 people are dying today with heart failure due to the lack of heart pumping functionality, and their choices are very few. So the goal of this product is to provide a better metric for us to better manage anticoagulation so these pumps can be used in more patients for destination. What we did was we took a peptide that had been used actually in clinical trial but failed as a contrast agent for MRI, and we redesigned the whole probe around it in such a way that we had 4 arms rather than 1 or 2 that would bind tenaciously to a clot, and that we had 4 different places for a nuclear molecule atom called technetium, which has a very short half-life, to be coupled to it. It turned out that this probe bound to fibrin almost irreversibly. Trying to compete it off has been almost impossible, not only with just one of the monomer legs at a ratio of 120,000 to 1, if you will, but even the forearm itself without the technetium will not compete off the forearm probe with the nuclear label, which is amazing but critical. It means that under the high flow of the LVAD, this can bind. To date, the most convincing data has been ex vivo data, although we've done these types of studies in rodents. We have shown that unlike other probes that only bind to newly formed clots, these bind to clots that have been formed and aged in the body for between 7 months and 2 years. So it's able to bind these almost tenacious concretions, if you will, of fibrin and stick to it irreversibly for all practical purposes, while the blood level actually falls very quickly because most of the other probes circulates for days and weeks. It prolongs the time of the probe in the blood beyond the time that it's even radioactive. So we would never have a signal that we could detect. But ours, because of the way we constructed the forearm, actually goes right out through the urine and doesn't collect in the liver or spleen, which are organs close to the LVAD. This probe, because it binds to aged clot, where all the other probes really only bind to acutely formed clot, could bind to pulmonary embolus. So the CDC numbers for pulmonary embolus, patients reporting to the emergency with symptoms consistent with pulmonary embolus, are 700,000 a year. Our approach to tackling the pulmonary embolus market is to first get the IND and demonstrate the efficacy of fibrin detection with the LVADs, and then expand the IND to include other patients who now have a second indication, that be pulmonary embolus. The phase 1 study would be done in patients who are getting elective pump replacement because they're known to be or suspected to have clot. So we would do maybe a dozen patients, and then we'd go to phase 2, and we would probably do it on a couple different instruments in maybe 10 to 20 more patients. I think in 3 years, it can be manufactured and used in patients. A CEO, in the case of a small company like Capella, has to wear multiple hats, but they're mostly of the financial business development. So the CEO is responsible for the business itself and needs to focus on, 1, acquiring funding to do the $5 million step from where we are to the clinic. 2, to get that money from credible board members who actually can help to advance the company. They need to be people who have their own connections, that have their own experience in the industry.